I just wanted to give you an update about the Intergalactic Workflows Commission, or maybe you don't know it, so yeah. And basically, I want to ask you to join us, like maybe user workflows, or yeah, join the team and submit some workflows. Yeah, what's the IWC? So we're a group of workflow aficionados, I would say. It's centered around Galaxy workflows, and we started. Let me do this. Oh, that's easy. Much better. Yeah, so we started in. We had this idea in 2018 that instead of having all the workflows on, just a Galaxy instance where you can share them, we wanted to have them in GitHub and tested. And that was in 2018. Nothing happened until, yeah, right in COVID times, we actually got around to doing it. The goal is to lower the barrier to workflow use, make sure that sharing and reuse of production-ready workflows becomes the standard within the Galaxy community. We want to help with workflow development and testing. And it's come a long way since then, but yeah, there weren't any like established practices yet. Of course, we also transmitted the requirements to the development team, and we want to document tips and tricks and contribute to the training material. So where can you find these workflows? So this is at iwc.galaxyproject.org, and you may have seen that we've used this heavily also in the opening live demo session. Yeah, that's the entry point. You find a bunch of like workflows that we're proud of. And we have currently, I think, 103 workflows there in various different disciplines. So from computational chemistry to genomics, imaging. Yeah, I mean, and it's open to really any scientific discipline. I'm hoping that you might want to come and just, yeah, add your own workflows. So what do we have? So we have a page for every workflow. I just picked a random one out here, and we already mentioned like there's an about section. This is actually coming from the README of the workflow itself. We're encouraging sort of a bit of a standard approach where first you describe what the workflow does, what kind of inputs it needs, in detail what it's doing. And on the right side, there's a bit of like summary as well, and metadata like who's the author, which release is this, when was it last updated, the DOI. So you can get the DOI for every workflow submitted. We do this through Zenodo, a TRS ID, and there's a little widget for running the workflow at any instance you like. Yeah, we also have a nice little preview. It's a mermaid diagram that you can look around and see this workflow really what I want to do. Do the steps make sense for what I want to do? I mentioned versioning is very important and something that's a little difficult to do with like the embedded sharing feature inside Galaxy. But here you can really have like nice releases. These are actual GitHub releases in the back. So every workflow gets deployed to its own repository. So you can become the owner of that place and have your discussions there if you like. Yeah, and then, yeah, changelog is very important, of course, as you know, because you want to inform people what has changed in your workflow. Then we have a tab. I mean, here are the tabs. There's a how to run tab that we saw earlier as well. So it also has instructions for how to run a workflow on the command line, and on the command line that might either be on your laptop or in the demo we showed. You can also target your own cluster and yeah, pretend Galaxy is not a web server, but just a workflow executor. We have something cool that's run with sample data. So Actually, it's currently a bit of a misnomer because that's the test data that comes with every workflow. So if you select this and you enter any of the Galaxy instances that you want to use, we'll send you there with the workflow and with the sample data pre-filled. So that might look like this. I mean, we went over this really quick, but again, this is the description of the workflow that you can also see in the previous slide. Here is the test data, and you can see these are actually URLs coming from Zenodo. We want all our test data also versioned and accessible there. And then these are coming directly from the specification of the test. So both you can sort of run in the interface what we use automatically in our GitHub testing, but also it might help you to figure out what is actually valid input data. Yeah. So why would you want to submit a workflow to the IWC? There's a discoverability aspect. It's not hidden somewhere in the sharing section of a Galaxy instance where hundreds and hundreds of workflows are shared. You get a DOI for every workflow, or you can, of course, also come with your own DOI, and then we just link to that. We have automatic deployment of the workflows to use Galaxy, Workflow Hub, and Docstore. And in fact, when you click on run in Galaxy or run with sample data, we fetch the workflow from Docstore, and we store also that metadata there so that we know where the workflow came from. We have daily workflow tests. All the workflows have tests. They are executed in GitHub workflows. And yeah, as I mentioned, we do daily tests. We also make sure that when there is a new Galaxy release, we are not releasing Galaxy if it breaks any of the workflows. Yeah, we have automatic updates. I'll show that later. Versioning, I mentioned this. It's a central place also for bug reports. It's actually not heavily used, but of course you can open issues there and say that like maybe the result didn't make sense or it crashed. We want to start doing benchmarks, and yeah, you've seen the static page. So how do you contribute? Again, there's a contributing document, and also on the static page, there is a how to contribute link. And it has basically all the instructions to get ready. And it's a multi-step process, and I also follow this every time. And yeah, it should just work, hopefully. If not, let us know. Then you can open a pull request. Also, the pull request contains instructions for the reviewer, which, I mean, in a way, it's also instructions for the author, because the reviewer only checks it if you did those things. So for instance, we want the Docstore YAML file. Workflow is generic enough to be used with lab data. It's not just, following a tutorial or something. There's an annotation field. The inputs are human readable because this is, it's a user-facing thing. So everything should be human readable, not random uppercase, lowercase, dashes, underscores, what people like to do with command line centric things. Also, the workflow name is important. Yeah, a README that explains everything. I mentioned these things. And we want small files for the test cases. The test should really only exercise that the workflow functions correctly, but not like, it's not benchmarking. It really just makes sure that the workflow executes correctly. I mentioned we have automated tests, and then you have either passing or failing report, and you can inspect the results in detail there. And we also have automatic updates. So whenever a tool that is referenced in the workflow is being updated independently, the bot will check every week whether there are updates to be done, and then it will automatically propose the updates. The tests will run, and when they run, you can just merge them and create a new patch version. Yeah, I mean, I mentioned we had this idea in 2018, but then the first workflows we only added in 2021, and we've seen steady growth. Particularly last year was a strong year, and we're of course not yet through in 2025. The co-fest hasn't happened yet, so maybe we can push the numbers here as well. And also, yeah, the number of authors per year is growing steadily. So I'm very happy to see this. Yeah, so how to join? There's a link here. Also this, I should have put the link again here. It's gxy.io/GBCC-IWC. We have a element channel, matrix channel. We have a video call every second Thursday. There's going to be the co-fest. Oops, that's from a previous presentation. Yeah, so any contribution is welcome. Yeah, with that, I want to say a big thank you to everyone who has contributed to workflow and to Danon and Ahmed who built the static site. Thank you.
Okay, hi everyone. Today I present a project that is something quite new for us, and we want to work with digital pathology and H&D images. Images, there are several types. There are, for example, TC scan, MRI, immunohistochemistry, H&D, and we decided to focus on digital pathologies just to find out if there are some patterns, some features that we can extract, and also we can integrate with omics data. Also, this project is part of a working group in Bioconductor. The name is Multi-omics Integration of Histopathology Image Analysis. It started last January, so it's 6 months now, and this is one of the first, something concrete from this project. We focus on a workflow, and we decide to start because in Bioconductor, there are a lot of packages to deal with images, but not so much to deal with digital pathology and H&D images. So we decide to try with some example before, like the TCGA data. And we decided to use these data. There are 11,764 diagnostic images from 33 different tumor types, and they are saved in SVS format. Of course, in Bioconductor, there are several tools, several packages to deal with TCGA, for example, TCGA Biolinks, TCGA Utilis, and curated TCGA. But all this package just give you the chance to download the images and not to process them or do something or find a curated analysis. So to download all these images, we took 25 days and they are like 11 terabytes. So we need a lot of space to store them. And we choose these diagnostic images because they are the 40x. I don't know if, okay, the 40x just right there. So the quality is the highest one possible. And we want to do some feature extraction on these images and provide to the user the feature extract from the images, just something available in a package. The feature extraction, we use the human readable. For example, the human readable, a feature that has a biological meaning, and you can use them. We start using HoverNet, a deep learning model that we use for segmentation and classification. And with HoverNet, we did a nucleus segmentation, in which we classified the nuclei in benign, neoplastic, inflammatory, immune, necrotic, and stromal. Then the output is a JSON file, and inside this JSON file, we have the position of each nuclei and the label about this nuclei. Then we use the input, the JSON file from the previous HoverNet model to extract some feature with SqueakPy and scikit image, and we extract some feature related with a spatial feature, intensity feature. They has, as I said before, they has a biological meaning. Then also we are now working on QPath. And as first things, we have to convert the JSON file in GeoJSON. And QPath is very user-friendly, but sometimes you have some problem when you have analyzed images in batches, like you have multiple images, like 11,000. So, yeah. And the type of images that you can extract with QPath are, for example, morphological feature and the eccentricity, shape of the nuclei. So, they are quite interesting. And then also we have the embeddings. The embeddings doesn't have a real biological meaning because they are just number, and they are quite similar to the number that you have on TSNE and UMAP in transcriptomic analysis with your single cell analysis. And for doing that, we use ProfGigaPath. ProfGigaPath is a foundation model, and the input is a whole slide images, so we consider the entire images, and the output is a CSV file for now, but we are thinking to move it to Parquet or something more easy to use in R. And then all these features will be now integrated in our package. The package is image TCGA. This package is also created to provide a Shiny application to explore the TCGA data. and is on Bioconductor since April 2025. This is the graphical interface, so you can filter for the project ID, sample type, you see the total number of record, you can filter, so you will see the number of patients, and then you can see also the number of images that is different sometimes from the number of patients. If you use that link on the top, that is the same on the QR code, you can just go now and just play with the tool and the Shiny app. And then if you want, if you select your group of patient, you can click on generate our code and you have the code to download on the images on your computer. And also to validate the result, we are starting a case studies focused on ovarian cancer. For the images from ovarian cancer, we have 107 images. And also we want to integrate the information with RNA-seq data because the TCGA provide information for 429 images. And our main idea is to integrate this feature that we extract with TCGA clinical data, survival data, also using consensus OV, that is another bioconductor package, or genomic supersignature, just to try to understand if this signature can be helpful to find a pattern or biological meaning, something that can help scientists in cancer research. Also, we check the correlation of the purity extract from overnet because we compute the purity from overnet, like the number of neoplastic nuclei on the total number of nuclei, and we correlate with the correlation from the paper Aran et al., in which they provide absolute, lump, and estimate. There are three different correlation techniques based on somatic copy number, methylation, and transcriptomic data. And the result, the correlation is not so high, I have to say, but the p-value is lower than 0.05, so it's fine. So it does mean that probably the segmentation is working well, and we are just a first result, and maybe we should go on this analysis. And that's funny, I think, because it's the time that we spend to all this stuff. For the download, it takes like 1 month. For the overnet, 6 months. For Prof GigaPath, we should take 6 months. We are running now. For the SquidPy, 3 months. For converting GeoJSON in GeoJSON, 3 months. And for QPath, we are thinking about 7 months. So the total amount of time, it should be like 26 months. So it's like 2 years. But if you don't want to spend 2 years, you just download the package and you should have all the result in like a couple of minutes probably. And that's the status for the download the overnet. We're already done. And now we're working on Prof GigaPath, SquidPy, and the other part are more or less we're working on. And also we have another project that is TCAI images. It's another repository of public data. They also have different images, also the diagnostic one, but also radiomics. And they are saved in different formats like TIFF, SVS, or PNG. And we didn't download all of them, but if you wanna check how much time you spend to download all the images, you can use TCAI API. That is another R package, is available on GitHub now. And thanks also to Marcel for creating this package. And yeah, you can do that and just let us know how much time you spend to download all the images. And so in conclusion, the feature will be available soon on Image TCGA. And also we are trying to amplify TCAI API with new function. So that's it. Thanks for your attention.
So we have time for maybe 2 questions. Here.
Thanks, Ilaria. Just a curiosity, actually. You talked about histopathology, but does TCGA hold any immunohistochemistry staining at all?
I don't think immunohistochemistry, no, but if you want, they have also TAC or X-ray, for example, MRI.
Oh, okay.
So, no. But they have 2 different types of diagnostic images, the 40X and the 20X. I can show you here. Okay, that was quite far. Okay, here on this D section, there are the 40X, that they are the one that we use, but also they are available the B section, the 20X. They are a lot, but the quality is not so high. So we decide for just now, just focus on the the one in which the quality is higher, just because sometimes when the quality is not high, you don't have just good result. So.
Yeah, just a comment. I think
Yeah.
talking about this, I think Luigi also is kind of working on this direction, but thinking about that, it would be very helpful resource to now people try to infer kind of cell types from these images, you know, a kind of pseudo histopathology, if you will. So linking those predictions together with these images could enable major studies. Just that.
Good afternoon, everyone. Thank you. That was a great introduction. So now I can make my talk hopefully on time now. So today I'm going to be presenting on behalf of Cameron Watson, Varshini Vijay, and Luke Sargent. We're all at the Knight Cancer Institute at OHSU. We've also been doing some histopathology work with deep learning models, and our goal is to both develop tools within Galaxy, but also come up with compelling use cases that we have with our clinical trials at OHSU to have, some interesting applications. So I'm going to skip most of this because I think you already saw this. One thing I did want to point out is kind of the types of models that we're seeing within the context of histopathology. Some ones we're familiar with is segmentation, both at the nuclei, but also at the tissue level. And then there's also some like substructure segmentation and classification models that are happening at the same time. There can be pixel or region level inference to other data modalities. But of course, what we really want to focus on for our use case today is the patch and slide level foundation models. So one of the things that we've noted, and I think others in the community too, is that the image pre-processing is a really important thing to consider as we're working especially with these public data sets. As you mentioned, the differences in magnification can be problematic, but also for the most part in our pre-processing, we're chopping up the images to be into smaller tiles or patches. And if those tiles then have some of these image artifacts, sometimes depending on the task, your model can start picking that up and think it's really important, but obviously we don't want it to be caring about the slide edge. Same thing, there's a lot of stain variability. And so we're very cognizant of the need for some sort of stain normalization across our images. So we've been using the Trident toolkit, which is out of the Mahmoud lab at Harvard Medical School. There's a few reasons for this. One is that it's a kind of nice generalized modular framework that handles a lot of the pre-processing. It's not perfect. There's other things we want to add to or still use from other tools. But it also captures a lot of the tiling and feature extraction components for our embeddings. So this is a tool we've just started implementing in Galaxy. What you're seeing here is the Trident segmentation, where we can then be able to choose different segmentation models. This would be at the tissue level to identify where our tissue is on the slide. And we can remove things like pen marks and some of the other artifacts. What it's missing right now is the stain normalization. And we're also wanting to kind of compare against other artifact removal approaches. And so the other tools that we're looking at in the community is from Grand QC and SlideFlow. And so we're hoping once we can kind of get this tool more polished, we'll start working on integrating some of those others. And this is just an example of what your input and output within this Galaxy tool might look like, where your input in the original, this is of a section of a cancer tissue where the cancer is shown more there in the darker purple and it's outlined with that green pen mark. We don't want that pen mark in there. And we also want to have an outline of where our tissue is. And so in the segmented and QC'd image, unfortunately, they both show up as green, but there's a lime green outline of our tissue, and the pen mark has now been masked out of that image. The next step of this is doing the image patch extraction. So originally we were using actually the Prov GigaPath. They have an approach for patching, and it was taking a really, really long time. The one in Trident sped it up infinitely more. It was so much faster. And so that's one of the other reasons we've liked this. But some other things to consider as you're doing patch extraction is maybe you want to not do just these like already preset tiles, but you may want to have like regions of interest. You may have objects or structural features in your tissue. And so we'd like to look more at tools that could also handle that instead of just having preset tiles. But the other thing is that it's important that Trident does cover is resizing the images. So even if your TCGA images are 40x, but your ones at your own institution are 20x, you can kind of downsample or rescale those images to then match what the model was trained on. And yeah, so this is the Trident patching, and there's an example of what that output looks like on the bottom. The other thing we like about Trident is they've incorporated 22 different deep learning models. A lot of these are vision transformers and foundation models that have been trained on tens to hundreds of thousands of samples. And for the most part, you can swap these in and out pretty seamlessly. We've had a lot of success, I would say, with about half of them so far, but we're still trying to work through that process. And this is also a really nice thing in Galaxy if you want to allow a user to kind of pick and choose whatever their model is, do some benchmarking against it. So we're still in the process of getting this going. Actually, the hardest part is creating Docker containers that actually have all of the requirements for all of these different models. So that's kind of one of the sticking points. But the other thing we want to include here within Galaxy is most of these models are on Hugging Face. And so we'd like to come up with a way to have better integration and communication across Hugging Face and Galaxy. So if you have already, got your token for Hugging Face and you were aware of the restrictions and licensing for that model, now you could have Galaxy pull that in automatically for you. And potentially we could even start doing things like smart caching so that maybe if the model's already been downloaded once, we check if the user has permission to use that model still, and we don't have to keep re-downloading. So hopefully it'll speed up some of the tasks as well. The other kind of model zoo we're considering is the NVIDIA MONAI. And the other benefit here, when we have this many different models to choose from, we can start doing this cross comparison of our patch encoders, our slide encoders, whether that's actually keeping the spatial context or kind of doing some sort of pooling of your patches. And then we can also take whatever our classification task is and try and either do fine tuning or freezing those weights and then just creating or training a regression model on top of that. And so right now, that's one of the things we're doing is taking a few of these really popular models and kind of doing this all by all, like swapping in and out and seeing what models perform best. I'll show in the next, our use case has been within TCGA. It's kind of our starting point as well. We're using the BRCA data set. One of the things we want to be able to use as a task is subtype classification. So to start with that, we've just been doing estrogen receptor positivity. I don't know if the images are available, but I do know they have if the sample is ER positive or negative. And so we've been able to use, I think this was one of the uni models and ABMIL that's shown up here. On the top is the ER positive, but we're taking that one step further and then also comparing to gene expression signatures. The one shown here on the bottom is the PAM50 subtype classifications for breast cancer so that we can break things down into luminal, HER2, basal, and normal. It may not be surprising that these actually both do pretty well because PAM50 has a lot of overlap with IHC markers, but not perfect. But where we really want to go is within our own use case is being able to predict drug response. And this is a hard task because even though there's a lot of H&E images out in the field, that doesn't always mean that you have the clinical covariates, the drug response, or even sometimes the other data modalities you would want to be able to train these models. We have a set of clinical trials that we work with in triple negative breast cancer where we do have response data. And so what we'd like to get to do is maybe if we're not having to do fine tuning of the model, but just taking the frozen weights and actually then building a classification head, seeing if we have enough data to actually just predict responders and non-responders. The data set's still very small though, so it's unlikely that's going to work. The other approach we want to do then is to take other biomarkers that have already been developed in the field that don't rely on the clinical covariates, but we know within our own studies have a high correlation with response. So the one you're seeing here is another subtype classifier. This is a classifier specific to triple negative breast cancer. And in our hands, the one subtype at the top, the BLIA, which is basal-like immune activated, if a patient is BLIA, they often have a pretty good prognosis on this trial. This is based off gene expression, so now we can open up the possibilities of actually finding more data sets in the public to do the training, the fine tuning, and then see if we can do that out of domain prediction in our own data set. And with that, thank everyone here. This is mostly the work of Cameron and Varshini. So thanks.
So we might have an online question. So, if anyone has the online question, so can you read it out loud for everyone to hear? Okay, so one question. So who has a question here?
Oh, that's a very cool talk. Thank you. So one of the technical concerns you mentioned was you have the different magnifications and need to downsample them. So is that something that the model, do you have to do the foundation models need anything special to deal with that? Or, when they're doing their convolution reduction, are they sort of amenable to these changes of magnification? And, does that not screw up? I'm just kind of curious, how that side of things works to,
Yeah.
compatibility.
How we're dealing with it, and I think this is how most others in the community are, though it may not be perfect. All of the models that were in that table, they actually say the magnification they were trained on. And so what we do is just try to match our other data sets to what the model was trained on. I don't know of anyone that's actually done the comparison of if you had both, how much the model would be affected. But I think it is a concern that most people have and would assume that you're going to get a drop in performance.
Hi. So I'm Nick Eagles. I'm from the Libre Institute down in Baltimore. And today I'll be talking about Visium HD data, trying to analyze it. And my goal is to get you introduced to the analysis possibilities with the Visium HD if you've seen it, but you haven't started with it. So there's a talk yesterday that sort of covered this, but some similar technology. So basically it's a spatial transcriptomics technology from 10x, very similar to Visium Standard and Xenium. Visium Standard being lower resolution, that should be 55 micron spots actually. And Xenium being single cell resolution, but generally less paneled genes. Visium HD is a subcellular resolution at 2 micron bins. And the data size is approximately 20 times larger in memory from tests I've done. So today I'll be covering some analysis steps such as QC normalization, batch correction, clustering, finding variable genes, and finding cells. So I'll be talking about what I'll call data forms. So basically the data comes in different resolutions, 2 micron being the largest, but then you can also kind of bin together expression to get 8 micron and 16 micron bins. So today I'll be talking about 8 micron size, the 2 micron size, and then actually individual cells. So first, the 8 micron analysis is when you don't really need the full resolution. So for certain things, you don't need the full resolution. So in terms of QC normalization, you can actually use the read 10x Visium wrapper function from spatial LIBD with a couple hacks. And on the right side here, we show on the top, it's a plot of the total UMI for one sample that we have. And you can see this technical artifact on the right-hand side. The bottom right plot here shows this sort of sliding window technique that I developed to try to find exactly where that boundary is and then filter out the array coordinates associated with that technical artifact. So you can read it in as a sparse or HDF5 matrix. So it's actually, the data is actually so sparse that it's not too big in memory as in sparse format. So you can't have it fully in memory. And then we do a very basic library size normalization because that's what actually, it doesn't blow up in memory like some better techniques. So one analysis you might be interested in is finding spatially variable genes. But before we do that, there's this SE raster tool and paper that shows that you can actually do this much faster and with less memory at a lower resolution. So SE raster actually lowers the resolution of your data essentially. And then you can run something like NNSVG to find your spatially variable genes. So on the right-hand side, we have 2 of the top genes for our data set. MBP being a major gene. GNG8 seems to mark this brain region since this is human brain data that we're interested in. But for certain analyses, you do want the full resolution and you can really benefit from that 2 micron scale. So in terms of working with the data at the 2-micron size, the QC and normalization is very similar. We still use functions from spatial LIBD and Scuttle. Something I wanted to highlight was this batch correction technique that we use. So essentially, we fit a linear model against the sample ID in our data. Very basic strategy, but and for chunks of genes, we residualize out the effective sample ID. And then that introduces a lot of non-zero counts when you previously had zeros. So biologically, that doesn't make too much sense. And from a technical standpoint, it really blows up in memory when the sparsity is decreased. So it sort of accomplishes two different things here. So it's that cleaning Y function from the Jaffe lab package that does that. So once you actually do this QC normalization and batch correction, then we can apply this amazing tool called Fixture that does clustering on the 2-micron scale. So at the top here, you can see one of our tissues, one of the brain tissues we have, and a little inset to show just the resolution that you're able to achieve here. And on the bottom, there's actually a trick you have to do to sort of trick it into thinking you're working with one sample when really you have 5. So you place it all on one spatial grid in adjacent sections with a little bit of space, and then you can jointly cluster multiple samples. So we've had to do some tricks and a lot of interaction with these authors to get this to work. So that's shown on the bottom, this little trick of combining samples. But I mentioned that Visium HD is subcellular, so naturally some analyses you'll want to do on individual cells rather than working with these square bins. So there's a couple different approaches you can take. Very recently, the 10x Genomics came out with this Space Ranger tool, Space Ranger segment commands that does a lot of what I'm going to be describing today. That wasn't available when I was doing these analyses. So I use this tool called bin to cell, which is pretty similar. They both use Stardist under the hood for segmentation. So what it does is it takes your H&E image and it segments cells on the image. And then it pulls together the counts from the bins that overlap each cell so that you get counts by cells matrix. So that's all actually in Python. But we convert the AND data to spatial experiment using some functions from Giotto, which seem to be more memory efficient than an alternative called Zell converter. And QC and stuff is done pretty similarly, where we can use spatial coordinates to filter out problematic regions like the ones I showed earlier. So once you do that with the cell level data, there's this tool called Banksy. You can also use it with, I think, Xenium and other data types, but also works with Visium HD. So they have a really excellent vignette showing how to do batch correction with Harmony if you have multiple samples. And then you can follow with the clustering. So on the right, we show some plots, and you can actually plot with some spatial LIBD functions, little small hacks with that as well, and happy to talk about that more. But yeah, this is a great tool for multi-sample cell level analysis here. And in summary, so I talked about a bunch of different tools. So spatial LIBD for reading in data and plotting, SE raster for lowering the resolution when you don't really need the full 2-micron scale data, and an SVG for finding spatially variable genes, Fixture for the very high resolution clustering on the original scale of the data, bin to cell for finding cells, and Giotto Suite actually does a huge number of things that I didn't cover, but I covered how to convert between Python and R objects, essentially, and bioconductor objects. Banksy for cell level clustering, and then I also mentioned some tips. So really use the minimum resolution that you need for your analysis. If you don't need the full resolution, you can achieve things much faster. And maybe it's not possible in terms of memory to do it otherwise. I talked about the trick to maintain sparsity when doing this batch correction linear model technique. So that enabled us to work with the really huge scale of the Visium HD data. And then some limitations I've noticed through trying out a lot of different software is that often things are developed for one sample and not multiple. So that may be a problem for our lab. I'm not sure how big of a problem it is for other labs, but and sometimes there's like non-default parameter choices that are not specified in vignettes. So documentation goes a long way in really making things clear for users. We also generated sort of like, we tried out a lot of different software in the field currently and made a little table showing like how long it takes to run, how much memory it takes, and whether it integrates with spatial experiment and stuff like that. So there's actually more tools, but these are the ones I've mostly discussed today. And with that, I'd like to acknowledge the teams that really made this possible. So collaborators at LIBD, where I work, as well as collaborators at Hopkins. So big team effort. Thank you.
So I may have time for 1 or 2 questions. Okay, here.
Great talk. I'm definitely going to hunt you down later. So what I've been looking for is with the HD is the cell and then breaking up by cells. So if you can do that, what is the point of going and doing it like at the 2 micron resolution rather than, well, that's the whole cell. We think pretty well we can tell it's the whole cell. Why would you do it at a low resolution or any other resolution, just then this is my approximate cell?
Yeah, great question. So I guess we're still ongoing in this analysis, but one thing we've noticed is that the segmentations for the cells are sort of capturing signal that's similar to like the nucleus more. So like we've lined it up with some single nucleus data and shown that that does a good job. But when you use a tool like Fixture that accounts for all of the bins, even the ones that are outside of cells, we notice that you can capture some biological patterns were captured there. So it seems like maybe there's more biology to be found if you don't restrict yourself to cells and like maybe extracellular environment type things. If that makes sense.
Yes, thanks so much. So we have seen a lot of or some talks about spatial, and scale is a very important aspect of spatial analysis. So I want to show you kind of the scale I'm interested in, which is multicellular anatomical structures. And this is motivated by the variety of anatomical structures we found in the brain. So these are histopathological images, and they should reflect a bit the variety of multicellular structures. And often biological function is reflected in the morphology of these structures and their location. So with the advent of spatial omics and spatial transcriptomics, this motivates data analysis approach that embraces these multicellular structures. And yeah, with our package, we try to kind of guide through different steps of an analysis. So to motivate it, I want to show 2 examples. And the first one is an example of an IMC dataset, so imaging mass cytometry. It's a spatial proteomics technology. And the researchers, they studied pancreatic islets in diabetes. And in course of diabetes, the beta cells within these pancreatic islets are getting destroyed, which they're producing insulin. So this is responsible for problems that are come together with diabetes. And the nice thing about this data set is that they profiled a lot of these islets. So here you can see one field of view, and we have the annotations by the authors. And so you see these islets, they form kind of very nice and regular structures. So it's nice for us to profile them. And we use a segmentation or reconstruction method that works based on point pattern density. That's very simple and it scales very well. So we can reconstruct these individual islets in the data set directly from the annotation in R. So we can do this for all the images and all the islets. And as I said, the nice property about these data sets is that it has a lot of images and a lot of islets to quantify. And then there's different aspects we would like to quantify, similar to what we have seen with H&E images. Here we do it really on the kind of the level of the cells from spatial omics technology. So we quantify morphology, we can also quantify metrics that are more related to biology. And once you have quantified a lot of things, of course, you will be interested into finding what changes between conditions. So performing multi-sample, multi-condition comparisons. And here, one thing that is very important with this type of data is to be aware of the correlation structure. Because if you think of the structure here, the islet is our observation of interest. We have often the case that we have multiple of these structures per slide, multiple slides per patient, and then multiple patients per condition. And this is very important to be aware of, because if you treat each structure as an independent observation and you do feature-wise linear models to see if there's condition-specific effects, then actually you can have a, like this is based on the data set that I showed. We performed null simulations, and based on the data set here, just the correlation structure that we observe, yeah, you can have a false discovery rate that increased by up to 20%. So an easy solution is using linear mixed effect models. You can account for different level of correlation. And yeah, then you stay below your default or your desired false discovery rate threshold. So that was the first example. And the second example I want to show is a bit different. I want to show a gene expression analysis that is aware of these anatomical structures. And for this, I switched to a Visium HD dataset. So a dataset that was just introduced before. Here I work actually on the 16 micrometer bin, so even larger. And this is the human tonsil, and all these small kind of blobs you see here, these are germinal centers. So the orange and the gray ones. And the germinal centers are quite important because they are the location where B cells are maturated. And they do this, the B cells do this in kind of a directionality. They pass through a germinal center from the gray zone to the orange zone. So as you can see here, we have multiple of these germinal centers. They all have a bit of a different orientation, a different size. Yeah, so when you do analysis, you might want to account for this heterogeneity and don't treat the whole slide as a homogeneous region. So what I did here is I used our tools to segment the individual germinal centers. And then I used information about the coordinates of these two different zones to perform to construct a reference axis through each germinal center, and also find an anchor point from which that is based to like at the orientation of one of these two zones. And then you can use this information to perform gene expression analysis that is aware of these gradients. And since you have gradients that are different for each germinal center, you can do it per germinal center in a kind of a meaningful way. So here I show it for one sample. What I did is I binned kind of the gradient into different bins, and then I pseudo-bulked within these bins. That helps with the sparsity of the data. And then I used information entropy approaches to find gene expression that is changes along this axis. And actually we can find some markers that are previously known that corresponded to different zones in the germinal center, but we also find a lot of different interesting genes because we have the full transcriptome data set from Visium HD. So yes, to summarize, yeah, I think structure-based analysis is very interesting because, yeah, it's inspired by this relationship of morphology, function, and location. And challenges are that, yeah, there is a large variety of anatomical structures. They have different complexity. Here is an example of a colorectal cancer dataset. So this is epithelium in the colon. And as you can see here, everything is a bit more complicated. Reconstruction of these individual epithelial structures is way more challenging. The other big challenge is that the feature space of things you could quantify is huge. So it helps a lot if you have kind of some biological insight or if you know a bit what to look for, because yeah, then you can set up a statistical testing framework, account for correlation structures in your data, and find interesting results. So yeah, we have a package, SOSTA, tries to help with some of these steps in Bioconductor. So with that, I want to thank everyone involved in this project, mostly Mark, my supervisor, and my lab, and yeah, everyone else. And yeah, here you can find the package. Try it out. If you have any issues, write to me. I'm happy to sort it out and have users that use software. And yeah, I think I have time for some questions.
Yeah, so I think we may have time for 2 questions.
Amazing presentation. I was wondering, you mentioned mixed linear models. Which is your package doing that, or which framework did you use?
No, I mean, there's plenty of methods, or you don't need to slide them. There's plenty of methods in R available, right? So we don't reinvent the wheel. It's just my pointing out kind of which level is important. And there's also measures to measure like the correlation at different levels in your data set.
So yeah.
And so we use LME4.
But for example, using LME4, do you kind of do these eBays? You kind of shared information between genes to find the variant structure, or how do you?
That's a great question. Yeah, that's a bit the challenge, right? Because we have actually the feature space is so vast. We have different things like proportion of cells is on a different scale than the area or something. So sharing like variants between different observations is a bit tricky in these cases. So currently we're exploring a bit like what are good things, and maybe also if you could like look at afterwards like group observations into groups and then account a bit for correlation within after testing if you do it feature-wise linear mixed effect models. But that's
just to mention the last thing, we developed a package if you need for compositional analysis that can do mixed model.
Ah, nice, yeah.
Is in Bioconductor. It's called SCComp, and
okay,
if you need it, we can.
Yeah, I will talk to you about it. Great.
Okay, maybe one tiny question. 15 seconds, but
fascinating. Two questions. One question: have you thought at all about the annotation of these structures that you detect, or aware of any resources or tools that would help with the annotation? I guess mostly it's manual, but one could think similar as for, like cell type annotation, that you take some morphology features or cell type composition and help with some sort of automated annotation. First question. Second question, you could do the same thing on a subcellular level, right? Where you maybe have like molecule level data, and you could find some interesting co-localization or structures of molecules subcellular. I wonder whether you have thought about this.
Yeah, that's two great questions. I think the first one is, again, like what I mentioned with features that you can quantify. It's the same for annotation. You have the space of the structure is so vast. So you have like, it's good if you have like some biological prior, like, the islet cells are the things that you're interested in and so on. So it will be interesting to think about ways to do it maybe a bit more in an unsupervised way. That's the first one. And the second one was subcellular. Yeah, that would be really interesting. And I think there's some work where like spatial proteomics has become very good in subcellular, and yeah, definitely could do it. So it's more a bit of an idea what you could do, and I think very general. So thanks a lot.
Thank you for having me. So today I'm presenting a work we've been doing to help people that assemble genome, accelerate the manual curation of the genome. So a bit of context, this work is part of a collaboration between the Vertebrate Genome Project and the Galaxy team. So we published last year, at the beginning of last year, set of workflow that have been used by the Vertebrate Genome Project to assemble the genome. And we are up to 238 genomes that have been assembled in Galaxy that are part of the Vertebrate Genome Project. And we're accelerating the process, improving the tool, maintaining this workflow, but the bottleneck to produce this reference genome is the manual curation. And we need to accelerate that because they want to sequence 70,000 species, so we need to go faster. So to help with that manual curation process and increase the speed of production of this genome, we're doing everything we can to automatize this process. So the manual curation, the goal of it is to identify misassembly, identify what scaffold correspond to what chromosome. And the way we do that is, is there a laser? No. Oh, this one, sorry. Right, so we have this high CMAP, and it allows us to see which sequence are close together. And so what we want to see is this nice diagonal where we have the scaffold and relationship between the scaffold and the same scaffold in the other haplotype, but no noise or no like broken scaffold. And we've been using dual curation, which means we map the read on the two haplotypes at the same time. And this has several advantages. First, it's easier to see misassembly. For example, you can see here, well, my scaffold should be straight, but I have actually a break. It's also easier to identify sex chromosome. If you have a heterogametic individual, then you can see, okay, this scaffold definitely has no equivalent in the other haplotype. That's probably a sex chromosome. And it's also easier globally, it's faster to run, and it's been working well. And so what we're trying to see is like, do we have this kind of weird behavior? And we're using pretext view, which is a software outside of Galaxy to kind of like move around the pieces and reassemble clean chromosomes. So in Galaxy, what we're doing is we're producing this IC map and we're adding a set of evidence to the map that helps us provide information, context information about the genome. So we have coverage information, telomeres, gaps, and gene annotation. And it's useful for several use cases. In this particular use case, you can see that instead of having a nice diagonal that go from one corner to the other, we've kind of like, this scaffold is broken compared to the other haplotype. And we need to reattach it, and we know where to reattach it because we have a gap exactly where this break is happening. So that's where we're gonna stitch the sequences together. Another use case is shrapnel. So shrapnel is all these like really tiny bits of sequence that you get at the end. And in that shrapnel, you have assembly crumbs that are pieces that haven't been able to be attached to any assembly. But you also have, in birds and reptiles, microchromosome, which are really small chromosome, but they are very interesting to study because they are usually gene-rich. And so we want to identify them. And that's when the gene are important to see, because if we see a bit that have like a lot of coding gene and have like maybe some telomere sequence in there, okay, we think that's probably a microchromosome, and so we're gonna extract it, and we need to be aware of it. So this is what the workflow looks like. I'm not gonna get into detail, but basically we annotate every haplotype separately, that we merge the GFF, we align the IC map here, we have coverage, telomere, and gaps, and all the inputs are whatever is produced by the workflow we published last year that are part of the assembly. So you just need the haplotype, IC data, and PacBio data. And it works for haplotype, diploid assembly, but we also have an option where you can, if you have just a primary and an alternate and you don't have two full haplotypes, you can then have the curation for only one of the haplotype. So it goes a bit faster, but we can still do more. So the next step in this work of helping with the curation is also producing a JBROS instance in Galaxy with the same evidence, the gene, the telomere, so you can zoom in a sequence and see, okay, exactly what's happening in that context. And we're also adding synthetic dot plot with related species. So you can see, okay, my scaffold 1 has genes that are similar, like the synthetic with chromosome 1 in a close related species. So maybe that's my chromosome 1. So these are all this information that are useful to get to the chromosome level assembly. And so this pretext map, once they stitch everything together, it produces a file called AGP, but it's not done. Once you have your AGP, you need to verify that everything is oriented in the same way. You need to verify that there hasn't been any obvious mistake in the assembly. And so that's the next step, having everything after the curation cleaned up and processed, so it's ready for submission. So you can find this workflow and all the one that has been published previously in the IWC that Marius presented before. And
we're still working a lot on it, so keep an eye. There's very regular updates, so things are moving. But yeah, all the updates, every changes is documented in IWC. And we also have training coming on the annotation, the creation process. And I want to thank everyone that's been involved in the project, both on the VGP side and on the Galaxy side. There's been a lot of progress to help us get this project going. So thank you, everyone.
All right, we are a bit behind. If no questions. Any questions? Maybe just one.
Great talk. So I have a question for the Galaxy genome assembly workflow. How customized is that to vertebrate genomes? And would it work for invertebrate genomes, probably, but not plant genomes? Or how would that?
It would work for any diploid genome. All the databases are the output are exposed, so you can change. We're using vertebrates for BUSCO or like for any QC, but you can change that to any organism you want. Yeah, as long as it's diploid, we've had people working on mosquitoes that are using this workflow, so